Have you ever wondered how a single cell can hold the instructions to build an entire organism? The answer lies within the fascinating world of DNA and RNA. DNA, or deoxyribonucleic acid, is often referred to as the blueprint of life. It contains the genetic instructions needed for an organism to develop, survive, and reproduce. These instructions are passed down from parents to offspring, ensuring the continuity of life. Understanding DNA and RNA impacts our health, the food we eat, and even the choices we make in our daily lives. RNA, or ribonucleic acid, plays a crucial role in carrying out the instructions encoded in DNA. Think of DNA as the master plan stored securely in the cell's vault, and RNA as the messenger that carries copies of these plans to the construction sites within the cell. Together, DNA and RNA orchestrate a complex and elegant molecular dance that underpins all life processes. This dance involves the replication of DNA, the transcription of DNA into RNA, and the translation of RNA into proteins. Get ready to unravel the secrets of these remarkable molecules that hold the key to understanding ourselves and the world around us. Imagine a ladder twisted into a spiral. That's the basic structure of DNA, the famous double helix. Each rung of this ladder is made up of pairs of chemical building blocks called nucleotides. There are 4 types of nucleotides in DNA: adenine, A, thymine, T, guanine, G, and cytosine, C. These nucleotides always pair up in a specific way, A with T and G with C. RNA, on the other hand, is like a single strand of this ladder. It also consists of nucleotides, but instead of thymine, T. It uses uracil, U. While DNA is like a stable archive, RNA is more like a temporary copy, carrying specific instructions from the DNA to the protein-making machinery of the cell. DNA provides the blueprint, and RNA acts as the messenger, carrying out the instructions encoded within the DNA sequence. These instructions ultimately dictate the production of proteins, the workhorses of the cell that carry out a myriad of functions essential for life. Think of DNA as a language written in a 4-letter alphabet. Easy one, C, jury, D, A, T, G, and C. A gene, a fundamental unit of heredity, is essentially a segment of DNA that carries the instructions for building a specific protein. Each group of 3 nucleotides, called a codon, codes for a particular amino acid. Amino acids are the building blocks of proteins, and their sequence determines a protein's structure and function. This flow of information from DNA to RNA to protein is known as the central dogma of molecular biology. The process of converting the genetic information encoded in DNA into functional proteins involves 2 main steps: transcription and translation. During transcription, the DNA double helix unwinds, and one strand serves as a template for the synthesis of a complementary RNA molecule called messenger RNA, or mRNA. This process is carried out by an enzyme called RNA polymerase, which recognizes and binds to specific regions of the DNA, marking the beginning of a gene. The mRNA molecule, carrying a copy of the gene's instructions, then travels out of the nucleus into the cytoplasm, where protein synthesis takes place. In the cytoplasm, the mRNA molecule encounters ribosomes, the protein synthesis machinery of the cell. Imagine you have a very important instruction manual and you need to make copies of it. That's essentially what DNA replication is all about. It's the process by which a cell makes an exact copy of its DNA before cell division. The process begins with the unwinding of the DNA double helix, like separating the 2 sides of a zipper. Each separated strand then serves as a template for the assembly of a new complementary strand. Free-floating nucleotides within the cell pair up with their corresponding bases on the template strands, A with T and G with C. DNA polymerase moves along the template strand, adding nucleotides one by one, ensuring that the new strand is an exact copy of the original. While DNA holds the master blueprint, it's RNA that carries the genetic instructions from the nucleus to the protein synthesis machinery in the cytoplasm. This process of copying a gene's DNA sequence into a messenger RNA, or mRNA molecule, is called transcription. The enzyme RNA polymerase binds to a specific region on the DNA molecule, marking the beginning of a gene. RNA polymerase then moves along the DNA strand, unwinding the double helix and reading the nucleotide sequence. Once the entire gene has been transcribed, the mRNA molecule detaches from the DNA and carries the genetic message out of the nucleus, ready to be translated into a protein. The processes of DNA replication and transcription are fundamental to life. They ensure that genetic information is accurately copied and passed on to new cells, and that the instructions encoded in DNA are faithfully translated into proteins. These proteins, in turn, carry out a vast array of functions essential for life. Understanding these fundamental processes has revolutionized our understanding of life. It has paved the way for groundbreaking advancements in medicine, agriculture, and biotechnology. The journey from mRNA to protein is known as translation, and it occurs in the cytoplasm of the cell. This process is akin to translating a language. The nucleotide sequence of the mRNA is interpreted to synthesize a specific protein. Translation begins with the mRNA molecule, which serves as a template. The mRNA is read by ribosomes, which are complex molecular machines composed of ribosomal RNA, or rRNA, and proteins. Ribosomes scan the mRNA sequence in sets of 3 nucleotides, known as codons, each of which corresponds to a specific amino acid. As the ribosome moves along the mRNA, it brings in new tRNA molecules and links their amino acids together, forming a long chain that eventually folds into a functional protein. tRNA and ribosomes are central players in the translation process. Each tRNA molecule is uniquely equipped to recognize a specific codon on the mRNA through its anticodon region. Ribosomes, on the other hand, act as the site where translation occurs. They consist of a large and a small subunit that come together during protein synthesis. The small subunit is responsible for binding the mRNA, while the large subunit facilitates the formation of peptide bonds between amino acids. Without tRNA and ribosomes, the translation of genetic information into functional proteins would be impossible. The knowledge of translation has profound implications in biotechnology. By understanding and manipulating this process, scientists can develop new therapies, improve agricultural practices, and create novel biotechnological applications. One significant application is the production of recombinant proteins. By inserting a gene coding for a desired protein into a host organism, such as bacteria or yeast, scientists can exploit the host's translation machinery to produce large quantities of the protein. This technique is used to produce insulin, growth hormones, and various other therapeutic proteins. In agriculture, genetic engineering leverages translation to develop crops that are more resistant to pests, diseases, and environmental stresses. Advancements in understanding translation have paved the way for the development of mRNA vaccines, revolutionizing vaccine development. Our understanding of DNA has revolutionized medicine, providing unprecedented insights into the causes of diseases and paving the way for personalized treatments. Genetic testing allows us to identify individuals who are at risk for certain inherited diseases, enabling early intervention and preventive measures. By analyzing an individual's DNA sequence, doctors can identify mutations or variations in genes that are associated with diseases like cancer, cystic fibrosis, and Huntington's disease. This knowledge empowers individuals to make informed decisions about their health, such as undergoing regular screenings, making lifestyle changes, or opting for preventative surgery. DNA technology also plays a pivotal role in forensics and criminal investigations. While DNA holds the master blueprint, RNA is emerging as a powerful tool for treating a wide range of diseases. RNA interference, or RNAi, is a revolutionary technique that silences specific genes, offering a promising new approach for treating diseases caused by overactive genes. In RNAi, small interfering RNA, or siRNA, molecules are introduced into cells, where they bind to target mRNA molecules, preventing their translation into proteins. This targeted gene silencing has shown promise in treating diseases like cancer, viral infections, and genetic disorders. RNA-based therapies represent a paradigm shift in medicine, offering targeted and personalized treatments that hold immense promise for improving human health. The ethical frontier. Navigating the genetic revolution. The rapid advancements in DNA and RNA technologies have ushered in a new era of genetic understanding, but they also raise profound ethical considerations. Genetic testing, for instance, while offering valuable health information, also raises concerns about privacy, discrimination, and the potential for misuse of genetic information. The ability to edit genes using technologies like CRISPR-Cas9 has opened up unprecedented possibilities for treating genetic diseases, but it also raises ethical concerns about germline editing, which involves altering the DNA of embryos or germ cells, potentially passing those changes down to future generations. The ethical implications of these technologies are complex and multifaceted, requiring careful consideration and open dialogue among scientists, policymakers, ethicists, and the public. Striking a balance between scientific progress and ethical responsibility is crucial to ensure that these powerful tools are used for the benefit of humanity. As we continue to unravel the mysteries of DNA and RNA, it is essential that we proceed with caution and a deep understanding of the potential consequences of our actions. The genetic revolution has the potential to transform medicine, agriculture, and our understanding of life itself, but it is our responsibility to ensure that these advancements are used wisely and ethically. The Symphony of Life. From the intricate double helix of DNA to the elegant dance of RNA, we've journeyed into the heart of life's molecular symphony. We've seen how DNA, the guardian of genetic information, faithfully replicates itself, ensuring the inheritance of traits across generations. We've witnessed the remarkable process of transcription where DNA's code is transcribed into RNA, the messenger molecule. And we've marveled at the intricacies of translation, where RNA's message is decoded to build proteins, the workhorses of the cell. This intricate interplay of DNA and RNA is not merely a scientific curiosity, it's the foundation upon which all life is built. It's the reason we resemble our parents, why a single fertilized egg can develop into a complex organism, and why life on Earth is so incredibly diverse. Understanding DNA and RNA is not just for scientists in labs, it's knowledge that empowers us all. It allows us to understand our own health, make informed decisions about our families, and appreciate the intricate web of life that connects us all. A Future Illuminated. As we delve deeper into the secrets of DNA and RNA, we unlock unprecedented opportunities to advance medicine, agriculture, and biotechnology. Gene editing technologies hold the potential to cure genetic diseases, while personalized medicine promises tailored treatments based on our unique genetic makeup. Yet, with this newfound power comes a profound responsibility. We must carefully consider the ethical implications of genetic engineering, ensuring that these technologies are used responsibly and equitably for the benefit of all humankind. The journey into the world of DNA and RNA is a journey into the essence of life itself. It's a journey filled with wonder, excitement, and a profound sense of awe at the elegance and complexity of the natural world. As we continue to explore this frontier of knowledge, we can look forward to a future illuminated by the promise of scientific discovery and the hope of a healthier, more sustainable world. Join the genetic conversation. That's DNA and RNA, the fundamental molecules of life, explained in a nutshell. But this is just the tip of the iceberg. These fascinating molecules hold the key to understanding life itself. They're driving incredible advancements in medicine, agriculture, and biotechnology, shaping the future in profound ways. What are your thoughts on the power and potential of DNA and RNA? Share your insights, questions, and reflections in the comments below. Let's keep the conversation going and explore the wonders of genetics together. Don't forget to share this video with anyone who might find it fascinating and subscribe to my channel for more explorations into the marvels of science.